So one of the more challenging aspects of neuroanatomy is that it's taught in two-dimensional cross-sections, and clinically the brain is viewed in two-dimensional cross-sections. In fact, the only way you have to look inside your patient's brain while they are alive is through 2D cross-sections, either CT scan, MRI. So you're viewing the brain in 2D cross-sections, but you have to do the mental gymnastics to manipulate those cross-sections and view them in 3D, and to understand the 3D spatial relationships between the various structures. For some people that comes naturally, for others it's more difficult, and this video will accelerate that learning process because we're going to overlay the 3D objects with the 2D cross-sections. We're going to focus in today's lesson on MRI, but these same principles apply when you're looking at CT scans. The first thing when you're looking at an MRI is to figure out what type of MRI. Is it a T1 weighted, T2 weighted, or a flare, T2 flare? So what's the difference between these sequences? In a T1 weighted, the gray matter is gray, as you can see the cortex here outlined in gray. The white matter is white, and the water is black. In some cases, there'll be a digital post-contrast where the vessels are brightened digitally. That's one of the series on the MRI. And this is best for viewing the anatomical structures. The T2 weighted is reverse of T1. So the gray matter is gray, the white matter is a darker gray. So you can see this white matter in here is a darker gray, and the water is a bright white. What happens in a T2 flare, which is fluid attenuated inversion recovery, it's the same as the 2T, but the water has been digitally blacked out so that it's black, and that just helps with the dark adaptation of your eyes so that you can see the contrast between the gray matter here that's lighter and the white matter here. In the images that I'm going to show you going forward, we'll be looking at a T1 weighted MRI. The next thing you need to do when you're looking at an MRI is get yourself oriented in space. So we need to review anatomical directions, as these terms will be used clinically when referring to various slices in 2D. And the best reference for orienting yourself in anatomical space for the nervous system is to use the neuraxis. The neuraxis is a line that's drawn along what would have been the embryological neural tube. So if you remember from embryology, the brain starts out as a tube. The tube is up and down along what becomes a spinal cord, and then there's a number of flexures where the tube bends and folds. One of the major flexures is this mesencephalic flexure, and so the tube runs almost horizontally when you're actually in the cerebrum. So we can call this cerebral line, this longitudinal axis. The next step is to orient yourself in anatomical space. So to do that, let's review the anatomical directions. These terms are used clinically when referring to a slice as being either rostral or caudal, superior or inferior, so we need to make sure we know all of these terms. Now, why am I spending all this time? Well, that's because the terms dorsal and ventral, rostral and caudal are in reference to the neuraxis, or relationship to the neural tube. So dorsal is the top or back, like the dorsal fin of a dolphin. Ventral is the belly. Rostral is towards the rostrum, the beak, or like the nose. So if you're along this neuraxis and you're moving in this direction, let's say you're looking at a brain slice here, and then you say, I want to look at a brain slice that's more rostral, you would look at a brain slice up here. So you're moving towards the beak. Caudal is towards the tail. So now these terms, dorsal, ventral, rostral, caudal, are in relationship to the neuraxis, which is nearly horizontal in the cerebrum, but nearly vertical in the brainstem and spinal cord. Now, the terms superior, inferior, anterior, and posterior are terms with relationship to anatomical space. So, as the human is standing in anatomical space, and these are in the world. So towards the sky is superior, towards the floor, inferior, towards the front, anterior, towards the back, posterior. So when you're in the brain, the term dorsal is synonymous with superior, but when you're in the spinal cord of the brainstem, the term dorsal is synonymous with what? Posterior. And that's because of the flexure mesencephalon. These terms have to do with relationship to the neuraxis. These terms are independent of the neuraxis. They're in anatomic space. And you need to make sure that you're familiar and can use all of these terms. So let's review planes of section as well. So here's some of these slices. So here is a sagittal. In this case, it's a mid-sagittal slice. So here's the nose, you can see. So this is anterior and posterior, superior and inferior. This is an axial slice. Now if you're cutting a brain on a table, it's called horizontal in the event that you hear this term in the wet lab. That's just another term. But radiologically we call it axial. And then this slice here, like cutting a loaf of bread, is coronal. Okay, let's practice. So we're going to go through the structures on an MRI, the ventricular system. We're going to look at the basal ganglia, gray matter structures, thalamus, hypothalamus, hippocampus, amygdala, the fornix, corpus callosum, internal capsule with white matter structures. We'll look at just the major brainstem structures in this lecture, and then the lobes. You ready to go? So here we're using the VHD, which stands for the VH Dissector Pro software that's provided by Touch of Life Technologies, and we'll provide information at the end of this video to how to access and use this software on your own. In this case, on the left-hand side, we're seeing the axial, the sagittal, and the coronal slices. I'm going to move the cursor back and forth to be able to slice through. So we're looking in this slice, in this enlarged view of the coronal slice here, and we can slice from rostral or anterior to caudal through the brain. Now, when I was learning this, I remember the instructor would always say, okay, here are the ventricles. There's the two lateral ventricles. Here's lateral ventricles here. And then they'd go back farther and they'd say, and if you go back more caudally, you can see the third ventricle. And then if you go back to the cerebellum, you can see the fourth ventricle. And it's always such a mystery to me, how on earth can you see the ventricles in like 1, 2, 3, 4, 5 places? Well, all we have to do is add the ventricles, and here they are. So we're looking at the coronal slice with the ventricles in place. So you can see the lateral ventricle. The lateral ventricle has an anterior or frontal horn, a posterior or occipital horn, and an inferior or also called temporal horn. This is the third ventricle. And then you can see here's the cerebral aqueduct that drains into the fourth ventricle. So next I'm going to add the thalamus, because the thalamus is a structure which is a major relay center for all sensory processing, motor relay stations, and limbic circuits. They all go through the thalamus, with the exception of the olfactory system, which goes straight into the limbic system. But the thalamus sandwiches the third ventricle. And in fact, you can see that the third ventricle is sort of donut shaped. But remember, the donut shape that you're looking at is negative space. It's empty space. Well, it's space that's filled with fluid, the CSF, right? And so that space is the interthalamic adhesion, where the right and left thalami adhere. So now I've added the thalamus, and I'm going to move the slice off a little bit so you can see it more closely. You can see the thalamus is this football-shaped midline structure that sandwiches the third ventricle. So when you are looking at a coronal slice, you're going to see the thalamus as this heart-shaped structure here. Now I can remove the thalamus so that you can see it, and you can see this dark structure here and here as the thalamus. Now one of the areas that students often confuse is the thalamus with the hypothalamus. We'll add the hypothalamus here in yellow. And the hypothalamus is a relatively small structure that has an incredible number of important roles for survival. Survival of the species, such as mating, and survival of the individual, such as the fight-flight response. And I'm going to rotate this so you can see the spatial relationships. you can see here that the thalamus is more superior and caudal, and the hypothalamus is more inferior and rostral. So when you're going through slices, coronal slices, rostral to caudal, you're going to slice through the hypothalamus first, and then you're going to slice through the hippocampus through the thalamus. But remember that they're both midline structures that are sandwiching. Now, two other structures I want to point out at this point here are the hippocampus, which is the gateway for the formation of memories. This is an important part of the Pez circuit. And you can see how hippocampus is on the medial and inferior edge of this inferior horn of the lateral ventricles. It's sort of being cupped, as if this were a hand, and it's being cupped by that. So it's shown here in purple. So we don't store memories in the hippocampus, but memories must go into the hippocampus before they form circuits through the hippocampus before they can be consolidated into long term. You can see here. Now, and then the other structure I wanted to point out is the amygdala. Now both the amygdala and the hippocampus are in the temporal lobe, and they're both important parts of the limbic system. As I said before, the hippocampus is an important component of formation of memories, and the amygdala is important for encoding emotions, particularly the emotion of fear. Now, if you look at the anatomical relationships, I'm going to give you an important clue here. If you're slicing through the brain coronally and you slice through the hypothalamus, which is a midline structure sandwiching the ventricles here in yellow, laterally in the temporal lobe, you should be slicing through the amygdala. If you are more caudal, cutting coronally through the thalamus, then you should look into the temporal lobe and find the hippocampus, right? So amygdala and hypothalamus are in line with one another. Thalamus and hippocampus are in line with one another. Let's look at the cross-section where we slice through the hippocampus, the hypothalamus, and the amygdala. You can see right here is the hypothalamus, and in red is the amygdala. And we're just seeing the most rostral tip of the hippocampus. Okay, but as we go further caudal, now the shapes look quite different. In fact, you can see thalamus and kind of more smashed and swirly hippocampus. The next structure I want to point out are the structures of the basal ganglia. Again, I'm going to use the ventricular system as a skeleton on which to build these structures. And the basal ganglia is an important system for proper initiation and proper selection of movement. So with disorders of the basal ganglia, there's not paralysis, there's just dysfunction in either in the initiation or the inhibition of movement. The three major structures of the basal ganglia include the caudate, the putamen, and the globus pallidus. And let's point those out now. Here is the caudate. It's lateral to the lateral ventricle. It's C-shaped structure that has a head, a body, and a tail, sort of like a tadpole. And there's two of them. There's one in the right hemisphere and one in the left hemisphere. Remember, when you're looking at an axial MRI, you're always imagining that you're at the foot of the bed looking up at the patient as if they're laying in a bed, their feet are towards you, and you're looking up into the brain. So this is the patient's right hemisphere, this is the left hemisphere. When you're looking at them coronally, you imagine that you're looking at your patient face to face. This is the right hemisphere again, this is the left hemisphere. The next structure is the putamen. I'm highlighting the putamen in green as well, because in reality the putamen and the caudate start out as one structure. They're separated by the internal capsule, which is white matter that splits them apart. As they're split, there's little striations here, and so together the putamen and the caudate are called the striatum. When you hear that in the clinic, somebody talking about the striatum, they're referring to the caudate plus the putamen. And actually the striatum is still connected in one spot. That spot is called the nucleus accumbens, and it's the connection here of the caudate to the putamen. It forms its own nucleus, and it's an important location for the dopamine in the reward and reward system. This is an area that's highly studied in research surrounding addiction. But please note that the nucleus accumbens is not considered part of the basal ganglia. It's part of the reward circuitry, but it anatomically is a place where the caudate and the putamen join. So the basal ganglia is the caudate, the putamen, and then the last structure is the globus pallidus. The globus pallidus, now we're looking up into the patient's brain, so we're inferior view, is medial and inferior portion of the putamen. Why don't we look at this from the perspective of the axial slice now? So here we're at quite superior. This is the blue line that we're cutting through, and we don't see the ventricular system yet, so that means we're not going to see caudate or putamen yet either. As we go more inferior, we're going to pick up the lateral ventricle, and lateral to the lateral ventricle is caudate. We can also look at this in a coronal view. So here we are very far rostral in the coronal view, and you can see the very tip of the frontal horn of the lateral ventricles. Lateral to the lateral ventricles, first little bit of gray matter that you see is caudate. And then there's the internal capsule in white and the putamen. You can see them now popped up. They are lateral to lateral ventricles. So lateral ventricles, lateral to that is caudate. Then there's the internal capsule, white matter piercing the striatum, and then lateral to that is the putamen. Do we see globus pallidus here? No. We're far too rostral in the slice. As we go farther back, we will pick up the globus pallidus. So watch the caudate and the putamen as we move from rostral to caudal. And there, do you see that lighter structure here? You can see now caudate, putamen, and globus pallidus. And you can see that it's not until we're pretty far down through the brain in a fairly inferior slice, axially, that we can see caudate, putamen, and globus pallidus. And along the midline here, sandwiching the third ventricle, what do you think that is right there? That's the structure we saw before, the thalamus. As we go more inferior, this is hypothalamus here. So let's put the thalamus and the hypothalamus on. So here the thalamus is in pink and the hypothalamus, we're just saying, so here you can see we have the caudate, the putamen, the thalamus, and the hypothalamus inside the brain. And we can slice it any way we like and see these structures segmented. The last structure I want to point out is the fornix, which is it's a white matter structure that connects the hypothalamus, shown here in yellow, with the hippocampus, shown here in purple. So we're looking at the coronal view right here, and one of the reasons it's tricky is that in the sagittal slice, you'll see the fornix as a midline structure. In an axial slice, you can cut it through in two places and see it here anteriorly and then posteriorly. It's sort of like the horns of a bighorn sheep. And this is an important part of the Pepez circuit, where fibers of the axons from neurons of the hippocampus bundle together to form the fornix, wrap around, and slice through the hypothalamus to synapse on the mammillary bodies. That's part of the Pepez circuit. If you're looking coronally, you can see the fornix as a midline structure just at the bottom of the lateral ventricles. If you're looking in the sagittal view, you can see the fornix here as a white matter tract just inferior to the lateral ventricles. So we have corpus callosum as that large white matter strip connecting right and left hemispheres. Then we have lateral ventricles, and then we have the fornix wrapping around. This coronal slice is a nice place to look at all the different white matter structures. So corpus callosum is a commissural tract connecting the right and the left hemisphere. The internal capsule is a projection tract going from the brain down to the spinal cord back up to the brain. And the fornix is an association tract. So far we've gone through the ventricular system, gray matter structures, white matter structures. Let's just review a little bit of the brainstem and the lobes. So here we are in a mid-sagittal view. If we go superior to inferior, we can see corpus callosum, lateral ventricles, fornix. Midline structure sandwiching this black area, which is the third ventricle, is thalamus and hypothalamus. And then we have the brain stem, which has three parts. This is the midbrain, this is the pons, and this is the medulla. The little backpack of the brain here on the dorsal part of the fourth ventricle is the cerebellum. And we can also see the lobes quite well. So frontal lobe, parietal lobe, occipital lobe, and then laterally, we don't see temporal lobe here because this is mid-sagittal section. This temporal lobe is going to be lateral. So out here you can see the temporal lobe. Here we are in an axial section. This is frontal lobe. At this point, because we're so far forward, if you see the slice where we are right here, we've cut through this blue line right here. We're not in occipital lobe. Back here, even though it's the bottom, is parietal lobe. And so that's something kind of tricky that I got to pay attention to. You don't see occipital lobe until you get much further down. Now we're seeing occipital lobe down here. You can see temporal lobe quite well here and here. And this hidden lobe inside, if you put your fingers inside the sylvian fissure and punch, you'll get another cortex deep inside the brain. That's called the insula, an area important for empathy and putting yourself in another person's shoes, and taste cortex is there too. Here we're going to look in the coronal view at all the different lobes. So here we're looking at eyes, so we're going rostral to caudal, so there's frontal lobe here. So as soon as you pick up the lateral ventricles and the caudate, you can start to see the temporal lobe. As we go further back, you can see caudate and putamen now, still the temporal lobe. Now we've picked up caudate, putamen, a little bit of globus pallidus. As we go further back, now you see thalamus and hippocampus. And as we get further back, this would be more of the parietal lobe, and further back is occipital lobe. Now let's look at it in a sagittal section. This is a parasagittal section. This is the hippocampus here, inferior horn of the lateral ventricle. This is amygdala up here. So hippocampus in purple, amygdala in orange. And those amygdala and hippocampus are in the temporal lobe. So here you're seeing temporal lobe, and as we slide in, you'll pick them up, amygdala and hippocampus. The last structure that is part of the limbic system is sometimes called the limbic lobe or the cingulate gyrus. This is this cortex that forms a belt around the corpus callosum. So this is all cingulate. So look at that. We've been through all of these structures here. Now let's review step-by-step guide of exactly what we did so that you could practice this at home. So remember, spatial relationships are key. The first step that I recommend when you're looking at an MRI of the brain is first figure out what type of MRI you're viewing. Is it T1, T2, or FLAIR? Or is it CT scan? Step 2, determine what type of slice you are. Is it a coronal slice, sagittal, or axial? And are you more rostral? Are you caudal? Are you superior or inferior? Next step, find the ventricles. Now, if something is being pointed to, you have to ask yourself, is the structure gray versus white versus black? And knowledge of the type of MRI you are will help you. And then you say, is this structure midline or lateralized? Remember, caudate is lateral to lateral ventricles. Putamen and globus pallidus are lateral to both. Internal capsule divides the caudate from the putamen, and that's what gives it this striated appearance, calling the two structures together the striatum. Remember that hypothalamus are both midline structures, and that amygdala and hippocampus are both lateralized structures in the temporal lobe. Hypothalamus and amygdala are more rostral, while thalamus and hippocampus are more caudal. So let's practice. So you can pause this and try to answer this on your own. The answers are from superior to inferior. Number 1 is the cingulate, also called cingulate gyrus, also called limbic lobe. Then we have the corpus callosum. 3, lateral ventricles. Use that as a skeleton on which to build all the other structures. Lateral to the lateral ventricles, number 4, caudate. Lateral to that, white matter structure. Number 6, internal capsule. Lateral to that, number 5, caudate. This right here, number 7, we didn't discuss in this video, but it's the anterior commissure. It's sort of like the corpus callosum for the temporal lobe, connecting these two lobes together. 8, midline structure, sandwiching the third ventricle, and we're fairly far rostral. That's the hypothalamus. If we're in line with the hypothalamus, that means that's amygdala right there, number 9. So we're more caudal now. Lateral to lateral ventricle is caudate. Lateral to that, 2 is putamen. Medial to the putamen, globus pallidus. What is this structure number 4? Well, first of all, this is a coronal slice. Here's the lateral ventricles. The structure is white, so white matter. It can't be corpus callosum. It can't be internal capsule. It's not the anterior commissure. This right here is the fornix. Now we're seeing things more clear cut. We're much further caudal than we were before. Number 1, midline structure sandwiching the third ventricle. Right? Your choices of things sandwiching the third ventricle are either thalamus or hypothalamus. And this is a big heart-shaped structure, so that's thalamus. In the temporal lobe, you've got a 50-50 shot. It's either amygdala or hippocampus. This one is sort of smushy, swirly. We're much more caudal, so there's no way this is amygdala. This is hippocampus. And then this cortex here that forms an island inside the brain, number 3 is insula, or the island of cortex. Answers are 1, cingulate gyrus, 2, corpus callosum, 3, lateral ventricles, 4, now this is another white matter structure. So see, I said, okay, this is gray, so that's a cortex because it's surrounded by gray matter. That's cingulate. This is white, so that's corpus callosum. This is black, oh, that's got to be lateral ventricles. Inferior lateral ventricles, white matter forming an arch, oh, coming from hypothalamus wrapping around, oh, that's got to be fornix. And then this is that football structure in the third ventricle, that's thalamus. 6 is pointing to black, so that's the third ventricle. But you would have the hypothalamus on either side of this. And then 7, 8, and 9 are parts of the brainstem, so midbrain, pons, and medulla. Last one, number 1, so this is an axial view. I find the lateral ventricles first. Then I find, oh, this is a gray matter structure, lateral to lateral ventricles. This is a T1 weighted MRI. That's the caudate. Number 2, internal capsule, sort of a boomerang shape in the axial slice. Number 3, putamen. And now another big round structure sandwiching the third ventricle.